I'd like to begin today's discussion with a little bit of a definition of model organisms and a note about scientific names. So a model organism is an organism that serves as a study system for a broad array of different biological processes and different concepts. And it's tricky to draw a clear distinction about when an organism becomes a model organism and when it's just simply a laboratory organism. So we're going to draw a couple of distinctions with some of these that maybe would not be classified as model organisms. They really need to have a huge body of work to be formally classified as a model organism. And one of the cool things about life on Earth is because of shared ancestry, all organisms have things that are in common. Some organisms have a wide, vast array of things that are in common, others have less or so. So whether you are studying a bacterium or a plant or a mouse or a fruit fly, all of those are going to have elements that are in common with any other living organism. So if you're interested in human health, there are some things that you can do in a bacterium, like E. coli, because it's very easy to work with, it's relatively inexpensive, you don't have lots of ethical considerations. So if the processes are similar enough in E. coli, you can use that in lieu of a more expensive organism or an organism where there are many more ethical considerations that need to be accounted for. Okay, so that's a model organism, and I'm going to give you about a half dozen different model organisms that have been used. Some of them are more foundational, like for genetics, and not necessarily evo devo, but they provided a system where the foundations for evo devo could be worked out. So we'll talk a little bit about what each of the model organisms was used for. Now, quickly, before I move into those model organisms, scientific names, typically we use a binomial, the genus and the species, or some combination. And a lot of times it's just convenience. So we almost always say E. coli. Coli is the specific name for the species, refers to the fact that they're found in the colon, in the digestive system. The first letter here, which is the generic name or the genus, is Escherichia, but it's kind of a complicated, I believe it's Greek in root. It's a big long name, so just by practice, we just say Escherichia rather than saying Escherichia coli, we just say E. coli. And so you'll see that sometimes we use the genus name, sometimes we use a abbreviation of the genus and the full species name. Other times we just use the common name, and sometimes the common name is very similar to the genus and species. So I'll point those out, but I just want you to be aware of that. And typically by convention, when you have a genus, it's capitalized. All other taxonomic levels above genus are also capitalized. And species, by a convention, is in lowercase. And then also when it's in writing, we put them in italics. So those are just conventions that are used, and you might see some people that break those conventions, but typically in scientific literature, you'll follow those and should be able to recognize those pretty easily and in a straightforward manner. Okay, E. coli is a bacterium. It's a gut microbe. It's very, very common. You have billions and billions of them in your intestinal tract. It's part of your healthy gut microbe. So that's part of a healthy human and many other species also, but a healthy human colon is full of E. coli. Now, you might, or if you had not had a scientific background, someone might be a little bit confused because you hear about E. coli poisoning, right, or E. coli contamination. And when E. coli is of a certain strain, there are strains that are dangerous, but by and large, the vast majority of them are commensals, and not even that, but mutualists with humans. They help us, they provide biological resources and services that our gut would not be able to do if we didn't have those E. coli. So it was just plentiful, it was easy to work with. One other word about model organisms. Sometimes the specific choice about which organism becomes a model is as much due to accident or just happenstance. Not that E. coli is better than maybe dozens or hundreds of other species of bacteria as a model organism, but it was readily available, easy to use, relatively safe. So that's why it became the model bacterium and not something else. There are many other bacteria that work just as well. Okay. There are many, many different lab cultures that have been adapted and modified, even now manipulated using modern molecular techniques to modify the genome. One of the most common ways that this manipulation, this modification is done is via the process of transformation. So I want to talk just a little bit about this because it's critical to our modern model organisms really across the board. It's more difficult to transform some other types of cells. With E. coli, it's rather simple. So transformation is a artificial, a lab-developed version of something that occurs very, very naturally. So in some strains of E. coli, they have the ability, and they have this unique external structure, so they're fairly easy to determine under the microscope. But these strains of E. coli are different because they have a little extra bit of genetic material, and we call that a plasmid. So a plasmid is a circular piece of DNA that can provide additional genetic instructions to the bacteria. So it can actually change its genotype, and if they're significant enough, can have a dramatic impact on the phenotype. Now, one of these types of plasmids transforms the bacteria into this type that creates these pili. That's plural. Pilus is singular. It's a long kind of a hollow tube that comes out and finds another bacterium, another E. coli usually, but occasionally even different species, and then puts a copy of this plasmid into that bacterium. So whatever characteristics are carried on that plasmid are now going to be transferred to this bacteria. Now in the wild, that often means that they then become an effective transformer, a bacteria that can transform other bacteria also. So this is called transformation. Okay, so that is a natural process. Now, there are other means by which genetic material can get from one bacterium to another. And whatever means is used, it is a form of horizontal gene transfer. So transformation is a form of horizontal gene transfer. In practice, this term transformation is often used for any sort of horizontal gene transfer. But to be really precise, there are actually 3 different types of horizontal gene transfer that can occur among bacteria. And the type that I outlined here that occurs naturally is more precisely called conjugation. Okay? So conjugation is when there's an active contact, a pilus, that connects the 2 bacteria. And genetic transfer is one bacteria gets some genetic material from a donor. The other way, which I actually don't have on here, let's go ahead and add that to this. You should know this one also, is called transduction. And transduction occurs when there is a viral mediator, a bacteriophage, which is a type of a virus that attacks bacteria. Now, typically, a small virus will attack a bacterium, hijack its genetic network, and make a whole bunch of new little viruses. Those little viruses go out and the cycle begins again. Occasionally, when this process occurs, a mistake is made. And rather than incorporating viral genome into the new virus, a little bit of the host genome is incorporated instead. And then you've got a fully functional virus, but instead of with infecting viral genetic material in it, it has a little bit of bacterial DNA. And so if that virus then goes on and infects a new bacteria, instead of now becoming an infected viral bacterium, it gets a little bit of extra genetic material that if everything is right, it can use in addition to its own genome. So that is the process of transduction. Okay, so conjugation is physical connection with a transfer of genetic material. Transduction is a transfer of genetic material via a virus. And transformation, which does occur naturally but is more rare, is a little bit of genetic material that is isolated, like a cell dies or somehow gets broken up and a little bit of genetic material is floating around, and a new bacteria takes it up without any direct contact and without any viral media, and that's transformation. All 3 of those are forms of horizontal gene transfer. And so we have laboratory forms of transformation that are very, very efficient. We do it artificially, and we do this by creating a plasmid. A plasmid is a circular piece of DNA, usually a few thousand, up to 10, 20, 30,000 base pairs long for some very big plasmids, but typically just a few thousand base pairs. And there are a number of characteristics of these plasmids. They usually are designed the ones that we use in artificial transformation, they occurred naturally before humans started using them. But the ones that we use are designed so that we can splice genes in and out of them, so that we can readily identify different portions of them. They often have promoters on them so that we can express the bits of genes that are spliced into it. And this process of creating a plasmid is often called gene cloning or DNA cloning because we are artificially making a copy of DNA. And the cool thing about this, and this is the great thing about model organisms, is because of the universal nature of the genetic code, whatever gene I put into this bacterium, into this plasmid, can be used by that bacteria. So I can put a human gene onto a plasmid, and if everything is correct and I've got the complete gene, that bacteria will then make copies, that is mRNA and final protein product, of that cloned gene. And in fact, we have many, many useful products that are made this way, sometimes for individual laboratory applications, but other times for very broad and useful human health applications. For instance, originally, all of our insulin by which we could treat a type 1 diabetes, I always get the two mixed up. I believe type 2 diabetes is adult, I'm going to double check here because I'm obviously not a diabetes expert, but I just want to make sure I don't lead you astray. So type 2 diabetes is, adult onset, right? Which can be caused by a number of different things, but basically means usually receptors are not finding enough insulin. So type 1 diabetes, which is also called juvenile onset diabetes, occurs because you don't make insulin. And so for both types, but especially for type 1, you can treat using insulin from a donor source. And originally that insulin for type 1 and sometimes type 2 diabetes, that insulin came from the pancreas of donor animals. So pig, cow, those were the ones that we had enough in slaughterhouses that we could isolate insulin from the pancreases of those animals. But other mammals are similar and close enough also. Their insulin is close enough to ours that it can take the place of our insulin. So originally, when people began to realize that we could treat diabetes, this is in the early 20th century, the only source for that insulin was from pig pancreas or cow pancreas. And so it was very expensive and difficult to obtain. But it was life-saving. Okay? And so people, as we began to realize the universal nature of the genetic code, people built a bacteria that makes human insulin. And they did this by cloning the gene for insulin into a plasmid, transforming a bacterium with that plasmid, and then just growing it up. You just put them in a happy environment with enough food, and they will just pump out human insulin, and you can then isolate it. And so we now have relatively cheap insulin from a transformed bacteria. Another general term for this is recombinant DNA. Recombinant DNA is any type of DNA from one organism that has been combined with the genome of another organism. Okay, so this plasmid, which is sometimes also termed a vector, that's the technical term before it's made into a complete plasmid. And there are all sorts of tools that we can do this. This is just one of many, many thousands of vectors that are available to make a plasmid. And they have antibiotic resistance genes on them. Those become tools for manipulation. They also have sugar digesting genes, which can also be a tool. We're not going to get into the details of this. In a future lab, I may outline it. I'm still trying to decide whether we'll do that in this new online version of the class. But for now, just know that there are several tools, and I'm going to list them here. The first are restriction enzymes. Restriction enzymes are proteins that will cut DNA at a very specific point. And so we have all these restriction sites. These are the different names for restriction sites that are built into this plasmid so that we can manipulate it and insert or even excise bits of DNA. It can be a PCR product, can be in a complete gene. Most of them are palindromic, but it's a very specific cut site where that protein will cut, and it won't cut at any other DNA sequence. So we can open up DNA and therefore ligate target DNA into that. So the process of making a plasmid for transformation usually involves a restriction enzyme to cut open the DNA, then ligation of a prepared piece of DNA with matching ends, sometimes they're called sticky ends, but matching sequences of DNA to then stick them into a plasmid. So once we have that plasmid, then we can transform the bacteria. And we transform bacteria in one of two methods. Either heat shock, so we get them from a very cold temperature and very rapidly heat them up to a little bit higher than they're comfortable at, and that shocks the bacteria and opens up pores in their cell walls. That allows the plasmid to get into some of the bacteria. And then they go back to normal, they have to recover, we grow them up and identify them, and now they are recombinant organisms that carry that plasmid with whatever piece of DNA we have inserted into the plasmid. The second method of transformation is electroporation. And electroporation works very similar to heat shock. It's usually a little bit more efficient, but the same thing. We shock them with enough electricity, not enough to kill them, but enough to shock them, opens up the cell pores momentarily, and allows that plasma to enter. Okay? Once we have done that, we have three categories of bacteria that are in our now transformed population. Okay? We have original bacteria that did not take up a plasmid, and that's probably the vast majority of them. We have bacteria that took up a plasmid, but the plasmid does not contain our gene of interest. So the process of inserting that bit of DNA into a plasmid is not 100% efficient. In fact, it's usually quite inefficient. So a small minority of bacteria in our population carry our plasmid and have our insert of interest. And these are the ones that we want, right? These are our recombinant bacteria with our target gene that we want to isolate from the other bacteria. But they're all floating around together, and so we need a method to distinguish these three types of bacteria. Okay? Now, the key to this, it's a two-step process. I'm going to outline, you should understand it in kind of big picture terms. The key to this process is the tools, these features that have been designed on the plasmid. Okay, so the first tool is an antibiotic resistance gene. In this case, this plasmid actually has two of them, but either one would work. So step one of the screening process involves that antibiotic resistant gene. So if we put these three types of bacteria onto a petri dish that contains an antibiotic, the wild type, the original bacteria, will die. So bacteria that do not get a plasmid die off. So that's step one of the screening process. So we get rid of the first type of bacteria. However, we still have two types that we need to distinguish. We have those with a plasmid without an insert and those with a plasmid that has an insert. Now both can survive in the antibiotic-infused plate because they carry the resistance gene on another part of this plasmid. But we want the ones with the gene insert. And so the second step involves a screening process. And for this screening process, we take advantage of this sugar-expressing gene and the fact that the insert site for our piece of DNA is right in the middle of this sugar-expressing gene. So if we do not insert the DNA, we have a complete sugar-expressing gene. And so bacteria that get a plasmid without a gene insert, they digest the sugar, there's a chemical change, and those bacteria turn blue. The ones that have a gene inserted into the middle of this sugar-expressing or sugar-digesting gene, they remain white, which is the wild type. And so we end up with something that looks like this, a plate with blue and white colonies on it. The white colonies are the ones that are candidates and most likely have our gene insert. Now, usually we'll do some sequencing, some PCR to have 100% certainty that they actually have the insert that we wanted, because there's still occasionally something that can happen. Okay, so you should be able to outline this two-step screening process once we have tried to transform a population of bacteria and how we get to the final white colonies that have our gene in them. And so we end up with a recombinant bacteria with whatever gene of interest we want. And this process of transformation is one of the major tools that has made E. coli such an important model organism. They are easy to transform, and we've done it millions and millions of times, sometimes for one specific laboratory application, sometimes for a widespread thing like insulin production, right? So that we can create. These processes are used when we're creating vaccines and really in just about any molecular lab at one point or another, they'll go through this process of transformation. The next model organism I'm just going to mention briefly is Arabidopsis, usually Thaliana, but again, Arabidopsis thaliana is a big long name, so more commonly people will just say Arabidopsis as the shortcut name. Arabidopsis is a plant from the mustard family. It grows very, very rapidly and makes lots and lots of seeds. And so this brings up one other important category or qualification for model organisms. We want model organisms to be easy to raise. We want them to be rather fecund, meaning that they make lots and lots of babies. We want to make them relatively easy to maintain in the laboratory. And so Arabidopsis fills those categories. And so, again, probably there are many other plants that would work just as well, but as a consequence of its characteristics and some just happenstance, it has been chosen. And so Arabidopsis is kind of the big model organism. It also has a relatively small genome, which wasn't known when it was originally chosen, but was convenient. It's been completely sequenced and studied and is completely understood. All the genes have been identified. And so you don't need to memorize all these details about it, but I do want you to understand why this makes it a good model organism. It reproduces very, very rapidly within a short time frame and makes hundreds of seeds for one individual plant. So it's a really convenient and great model organism to use. One of the other things that is useful about this plant is it can be transformed. The transformation process is a little bit different, and because this is not a plant genetics course and we're focusing on animals, we won't get into all the details, but just know that it can also be transformed, and so we can get genes from other plants into Arabidopsis so that we can study them and do that and research them. Well, maybe the only detail you need to know about this is it's mediated by a bacteria. It's called agrobacterium. So that bacteria it basically infects the plant and carries the new genes into that plant. And that's as much detail as you need to know about Arabidopsis. I've added a microscopy thing here. This really can apply to all organisms, but microscopy is a really important part for the study of model organisms also. It's just kind of a pretty picture, shows developing cells and some of the things we talked about in the last lecture about how we can differentially label cells that are physiologically different. Okay, our next model organism is Drosophila. And Drosophila has been the workhorse for genetics, genomics, many other areas for all animals. So if you need a model for animals, and bacteria won't cut the bill because there's some things that are unique about animals that bacteria don't do. But if you need just a standard animal model, Drosophila is the one you want to go to. It is very cheap and easy to maintain in the lab. It has a very, very rapid generation time, and we can also manipulate it. So again, we probably could have chosen several other species or even several other types of insects. They would have been maybe not quite as good. Drosophila is really well suited as a model organism. Okay, so males and females are easily distinguishable. They are dimorphic, meaning males look different from the female. So males have this. This is where the name for the species come. Melanogaster is the most common species used, and it means pigmented abdomen. Other species of Drosophila also now serve as model organisms, so we have a wide range to choose from. There are many mutations that have been identified, and in the early days, people would purposely expose them to radiation or certain chemicals that would increase the mutation rate, and then they'd look for those mutations, hoping to find something random. In our next lecture, we'll look at how Lewis was basically the first modern evo-devo researcher, and he used this process of randomly mutating fruit flies and then looking for mutations. So we'll talk about that in our next lesson. They also have these polytene chromosomes, which in the early days made the visualization of their chromosomes much easier because they were larger. Polytene just means you have multiple chromosomes kind of all sandwiched together. We can stain them. This is a stained one with differential stains, and we were able to identify different regions of the genome even before we could sequence the genome and look at it at the DNA level. I'm not going to make you read this paper. You can look it up if you're interested, but I won't ask you questions about specifically, but it talks about some of the many hundreds and hundreds of discoveries that have relied on Drosophila. So Drosophila is the key to much of our understanding of biology in general, and specifically biology of animals. So I'm just going to list a couple. Again, you don't need to memorize all these, but just know generally that these things were found because we worked with fruit flies. The idea of chromosomes, that our genome is organized into these different subunits that we call chromosomes. The patterns of heredity, which were again first discovered in pea plants, were then confirmed in fruit flies, and then the more complex forms of heredity, such as sex-linked chromosomes, much of the more complex genetics, not just recessive dominant, but more complex genetics. The vast majority of these were discovered in fruit flies and then confirmed in other organisms. How genes are organized and controlled. So I gave you that diagram of a gene at the very beginning of the eukaryotic gene at the very beginning of this class. That was all discovered in fruit flies. And then transgenetics or transgenic organisms, where we can transfer them from one organism to another, and how we do that in animals was first worked out in fruit flies and then later extended into other organisms. Now, I could probably make this list, and I believe the paper here does make it much, much longer. Many things that we just take for granted that you learn, maybe even going back to elementary school as basic fundamentals of biology, were first discovered in fruit flies, in Drosophila. Okay, so just know that if you want the most simple, basic animal model, that you're going to use a fruit fly. Okay, now I'm going to list a couple other organisms here that are probably not true model organisms because they really don't have the massive body of work, but that's kind of an arbitrary line. The reason I am mentioning them, even though they're not maybe true model organisms, is they played an important role in the study of development and evolution and development in Evo Devo. So Xenopus is commonly called the African clawed frog. Xenopus laevis is the full name, but Xenopus is most commonly term used. And these were used and are still used to some extent, but were heavily used in the early days of studying development and even into once we started doing Evo Devo at the genetic level, because they have large embryos and those embryos are visible. They have this kind of gel coating around them. If you've ever seen frog eggs, some other frogs do this also, but they have this gel coating. You can actually watch development occurring, particularly under a microscope. So this is at the 8-cell stage. But the cool thing about that is you don't need to fix, that is, add formaldehyde and kill the embryos to study them. You can just kind of visualize and watch them occurring. And there's an interesting resource here. I haven't checked. Let me double check and make sure this is still an active link. It's been a couple of years since I checked it. But you can see if you're interested. You don't need to go here and memorize or learn any of this. If we were in a more standard classical development class, if you've taken Dr. Booth's, then you may have done more about this. We'll learn a very basic amount of this, but you can watch this occur and you can learn about this at this website. And this is, again, all based on this work in a large body, so maybe moving into what we would call a classic model organism, but a large body of work that has made them important for Evo Devo. Okay, so just know them, and then if we wanted a model of embryological development for animals, the frog would be a really, really good choice because it's relatively easy. They make lots and lots of eggs. We don't have to kill the eggs. We can watch the process occurring. Okay, so it's a very important developmental model. The next one, which has also been important for Evo Devo, particularly development of the nerve system, is a fish called Danio. Danio is the generic name. Ririo is the most common species, so Danio ririo, but we'll just refer to it as Danio from here on out for convenience sake. It's called the zebrafish. They're actually also kept in freshwater aquariums just for pets, but they are done in a wide scale as a developmental model, a genetics model for a vertebrate. So they were a little bit easier to work for neurological development and genetics than the frogs were. So they've been widely used. They also have stem cells. And fairly early on, we figured out how to transform these guys also so we could insert genes. And so this is important scientifically, but there have also been some less scientific applications. So you can get what are called glowfish that have had a jellyfish gene or some other fluorescent gene inserted into them, so we actually are fish that glow in the dark. And these are simply laboratory models, and originally they were done because they needed a gene that was easy to tell if it worked. So they said, hey, let's make a glow-in-the-dark gene, and then if it works, they'll glow in the dark, and we'll know that we did it right. And then they realized, oh, hey, maybe we could sell these. So I think you can actually buy these. Sometimes they're available at pet stores or even like Walmart, but you can certainly buy them online. And they're just a genetically transformed form of the zebrafish. So know them as a developmental model that primarily are for neurological development. and some genetics and stem model work. Okay. Now, the last model organism that we're going to talk about is the mouse. And the mouse is really truly the only, well, I guess you can make an argument for the rat also, but those two really are the only true, what we would call true model systems. There's been a lot of work done in other systems, in guinea pigs, in primates, because they need to be done for something, but there's really not the amount of work that would raise them to the level of what we would call a true model organism. But with the mouse, they really have reached that status and were the earliest. Now, if you need an animal model, but you need one that is specific for mammals, which, there's some things like the immune system that is very, very different. So you can't use fruit flies to study the mammalian immune system, right? Because they have a very different type of immune system. But mice have a very similar immune system to humans. So we can use mice for many of the things that we can't do in humans because of logistics, because of expense, because of ethics, right, that we can do in the mouse, and they apply widely to humans. So this is the most closely related model organism to humans. Now, don't be mistaken. There are many other types of animals, the species of animals, that are much more closely related to humans. But because of price, because of logistics, they're really not true model systems. We use them as study systems, as laboratory animals sometimes, but not true model systems. So there are primates that are used sometimes when a mouse won't do the trick. But to do a primate study is orders of magnitude more expensive. There are more logistical and ethical considerations that you have to take into account. So by far, it's much more preferable to do it in a mouse well, it's better to do it in a fruit fly if a fruit fly works. But if that doesn't work, then you do it in a mouse. If the mouse doesn't work, and if it's an important enough study, then maybe we'll use something like a primate system to do the study. Okay. And then eventually, of course, once things have been proven safe and look okay, then we can transition to human trials that are much more tightly controlled and are much more stringent than any other animal laboratory system. But in the mouse, they have developed and found, some of them they've manipulated and made, but they've also found many strains of mice, and you can buy them now. There are hundreds of laboratory strains of mice that have either homologous or analogous processes to humans. Now, if homologous and analogous is not 100% set in your mind at this point, that's okay. I'm actually going to define it in much more detail in the next lecture, but hopefully you have some idea of what I mean here. So for instance, you can buy a strain of mouse that develops heart disease in patterns that are somewhat similar to what we see in humans. You can buy ones that develop neurological disorders that are related to some neurological disorders in humans. Not 100% identical, and obviously there are going to be some differences between humans and mice at any level, even if it's a homologous process. But these many different types of mice have made them an incredibly useful system for the study of many, many human processes. Okay. Now, I want to say one closing word before I finish up. This, again, is our last model organism that we're going to talk about, but I want to say one thing, and I'm going to do this in kind of a humorous way, but it's with a serious end result. So there's an article years and years ago that I found that was quite funny by The Onion. And if you don't know The Onion is a satirical news site, meaning they basically make stuff up for laughs. And they do it with a purpose, right? They're trying to satire has a purpose. They're trying to point out, flaws in our ethics maybe, or foibles in our culture, or things that are just silly or ridiculous. I saw one once that was about how a really bad basketball team, their stadium collapsed. And it really didn't, but, their arena collapsed. And the whole point of the article was how relieved the fans were because they didn't have to suffer through the awful torture that the game was because of how bad their team was. So that's the kind of sort of thing they do, right? Kind of poking fun at things. So this is one looking at mice as a model organism, and it's quite humorous. I'll bring it up here. You can read it yourself. I'm just going to highlight a few things. And there's some language here, so hopefully you're not offended too much by language. But nearly 700 scientists representing 27 countries convened at the University of Zurich Monday to formally announce that their experimentation on mice has been motivated not by a desire to advance human knowledge, but out of a sheer distaste for the furry little rodents. And this, it goes on to make up all these fake quotes. They've got this guy with a gross out face here. As a man of science, I deal with facts. And the fact is that mice are gross. They're squirmy, scurrying little vermin, and they make my skins crawl. I speak for all my assembled colleagues when they say that the horrible little things deserve the worst that we can dish out. Now, again, this is a humorous take, but the point is that there are ethical considerations, and they're kind of laughing and saying scientists are just these very vindictive, evil people that just want to torture creatures, and so that's the way they do that to mouse. Now, obviously that's satire, but there are some very quite serious impact for any model organism. And so as a wrap up, I just want to point that out, that there are major ethical considerations with whatever model organism we use. And as humans, for better or worse, we have decided that if an animal is more like us, then there are going to be more hoops that we have to jump through. And the I guess criteria is, are they suffering? But again, we're judging suffering based on our own maybe limited framework of how we decide suffering. Do fruit flies suffer when we study them? And I'm not going to answer that question, but there are serious questions there. Do mice suffer? And the idea is, yeah, they probably do. And so we try to be very, very careful about the methods and the materials. There are processes and boards of experts that you have to approve your research for if you're using mice as a study organism, less so if you're using fruit flies, and really none if you're using bacteria. And that's the decision we've made. If you're using primates, then there are going to be really, really major bars that you have to meet to prove that you're going to make sure the animals are not suffering too much and that the potential outcome of the research justifies whatever small level of suffering is going to be used. So again, this is a satirical article. Scientists don't really hate mice, and most of them are very responsible, and we try to balance the benefits and knowledge that can come from studying these model organisms with the inevitable impact to the organisms themselves. So just keep that in mind and make sure that you understand that there are ethical considerations that are usually very carefully taken care of, and most scientists are very, very good and responsible with doing that. And a lot of great things have come out of this.